Hey guys, this is Shula from shulatutoring.com, and in this video I will go over a practice test on cellular respiration. So just wanted to remind you guys how it works. So if you look at the description for this video, you will see that there is a link there. So if you click on that link, you will see a practice test on cellular respiration. So you can complete the test by just, looking at the screen and doing it on a piece of paper, or you can just download the file and print it. It's up to you. But I want you to complete the test first, and then you can watch the rest of this video, which is basically the explanation part. And as you are watching my explanations, you can see which questions you got right, which questions you got wrong, if any, and you can score your points and calculate your grade. And this way you will know how well you know this material. Okay, so let's start. First, we will go over multiple choice questions, 3 points each. Number one, so the first question, ATP hydrolysis is what? So we have 4 different choices here. Results in the formation of one organic and one inorganic molecule. B is exorganic. C releases energy that can be used to do work in the cell. And D, all of the above. So the answer is D, because ATP hydrolysis does release energy, which is choice C, but the definition of reaction that releases energy is that it's exorganic, so that's also correct. And ATP hydrolysis will result in the formation of ADP, which is an organic molecule, and inorganic phosphate, which is an inorganic molecule. So choice A is correct as well. So we have to choose all of the above. And then number 2, a large number of CO bonds would indicate that the molecule is oxidized. So it's choice B. So if you have a lot of CH bonds, that indicates that the molecule is reduced. Large number of CO bonds indicates that the molecule is oxidized. So if the molecule is oxidized, it has really low potential energy. So choice C is wrong. If the molecule is oxidized, obviously it's not reduced. And choice D has a lot of electrons that are not held very tightly and are available for chemical reaction. That describes a reduced molecule, right? Because reduced molecules have a lot of available electrons for chemical reactions. 3, during cellular respiration, glucose is oxidized, right? Because we are losing electrons. And that's what happens when we go through all those oxidation reactions and we break glucose down, right? So we start with 6-carbon sugar and we end up with carbon dioxide. So that's oxidation. And also, as we do that, we load electrons on electron carriers, and electron carriers use them later in electron transport chain. So all that is oxidation, and oxidation is defined as the loss of electrons. So glucose is not reduced, converted into molecules of ATP to be used for energy. That's not true, right? ATP is produced in a different way. Is phosphorylated? No, it's not true. The correct order for the 4 steps in cellular respiration in eukaryotes is choice A. First, glycolysis happens, right? And then glycolysis will result in 2 molecules of pyruvate, and then a citric acid cycle would take place. And after citric acid cycle, we would have electron transport chain. So choice A is correct. For 1 molecule of glucose that enters glycolysis, the net products are choice D: 2 ATP, 2 NADH, 2 pyruvate. So here, I don't think your teacher will ever expect you to know exact numbers of each type of molecule that will result from glycolysis. But what this question is checking is your knowledge that first of all, no FADH2 will come from glycolysis, right? They come from citric acid cycle, and that CO2 is also not released as a result of glycolysis, right? So ATP, NADH, and pyruvate, that's all that comes from glycolysis. Number 6, in eukaryotes, pyruvate processing happens in the matrix of mitochondria, because there is a big complex, enzyme complex, that does pyruvate processing, and it's found in the matrix of mitochondria. Nothing really happens on the surface of mitochondria, so choice A doesn't make a lot of sense. Cytosol, that's in bacteria, right? That's where pyruvate processing would happen, in bacteria. On the ribosomes, right, that's a different thing altogether. That's where translation would happen. For each glucose molecule, citric acid cycle will turn 2 times, choice B, because when we start glycolysis, we start with a 6-carbon sugar, which is glucose. And then we convert the 6-carbon sugar into 2 molecules of pyruvate, and each molecule of pyruvate will have 3 carbons. And then for each molecule of acetyl-CoA, citric acid cycle will turn once, right? So that's how we turn 2 times for each glucose molecule. Citric acid cycle will release choice A, CO2, NADH, FADH2, and ATP. All those molecules will come from citric acid cycle. Sometimes you will see ATP referred to as GTP, but that's almost the same thing. In regards to other answers, choice B misses the fact that CO2 also comes out of the citric acid cycle, and that's important. Choice C misses ATP, and then choice D mentions acetyl-CoA, which is the starting product of the citric acid cycle. Citric acid cycle doesn't release it. And then 9, ATP molecules are produced by substrate level phosphorylation during glycolysis and citric acid cycle. So choice D is correct. It's A and B. So substrate level phosphorylation is when we use enzymes. So choice D, A, and B. Here we see a reaction for cellular respiration, right? So we start with glucose and oxygen, and then we end up with carbon dioxide, water, and energy. So look at the reaction above. What happens to oxygen during cellular respiration? So what happens with oxygen is that it combines with hydrogen to make water. So this oxygen will turn into this. And the reason is that oxygen is a final electron acceptor. So at the end of the electron transport chain, it will combine with some protons and electrons to make water. Number 11, after electrons enter the electron transport chain, energy is released, right? That's true, because every time electrons go down one step in electron transport chain, they go down in energy level, and that energy will be released. Choice B, ATP generated during glycolysis and citric acid cycle is being used up. That's not true. Electron transport chain doesn't use up energy, it generates energy. Ubiquinone is not a final electron acceptor, so choice C is not correct. CO2 is released. That's not true. CO2 is released during citric acid cycle, not during electron transport chain. Most ATP produced during cellular respiration comes from electron transport chain and chemiosmosis. Other processes will release a small amount of ATP, but the majority of ATP will come from electron transport chain and chemiosmosis. An organism living in oxygen-poor environment, number 13, might use nitrate and sulfate as electron acceptors. Choice B is correct. So choice A will be limited to glycolysis for ATP production. That's not true. There are other chemicals that could be used as electron acceptors, even though they're not so efficient. So those organisms might not generate as much energy. So they won't necessarily be able to grow very big or produce very efficiently, but they'll they will carry out this process nonetheless. And then C will carry out glycolysis and citric acid cycle, but skip the electron transport chain. That's not true, because the whole point of citric acid cycle is that we need to load up electron acceptors with electrons to be used in electron transport chain. So it makes absolutely no sense to carry out citric acid cycle if you cannot do the electron transport chain. So choice C doesn't work. Nothing can live without oxygen. That's not true. There are organisms that are anaerobic, so they live in environments that are totally devoid of oxygen, and oxygen is actually poisonous to them. So choice D is not correct. Number 14, you feel pain in your muscles during exercise. The reason for it is that lactic acid is formed. That's correct. Electron transport chains shut down. That's correct. The rate of oxygen supply cannot keep up with fast glucose metabolism. So all of that is correct. So correct answer is choice D. So what happens is we need glucose and oxygen in order to proceed with cellular respiration and generate ATP. But when we're exercising really fast, then we cannot deliver oxygen at a rate to keep up with our need for energy. So that's why electron transport chains will shut down, and we will have to switch to fermentation, which means that lactic acid will form as a byproduct, and it will cause pain in your muscles. During alcohol fermentation, acetaldehyde will accept electrons from NADH and form ethanol. The reason it happens is because NADH must be regenerated for fermentation to continue. So why is that true? Because during glycolysis, we will load NAD plus with electrons. And in the end, those electrons will go to electron transport chain and they will be used there. If we cannot proceed with citric acid cycle and electron transport chain, then we run into the problem that we have NAD plus loaded with electrons, but they have nowhere to go. So the main point of fermentation is that we have to regenerate NAD plus, and then fermentation will be able to continue. And even though we won't generate a lot of energy, we will generate some energy, enough energy for that organism to live. So those yeast cells will be able to survive just with fermentation. A reaction that breaks down large molecules into small ones is called catabolic. Anabolic reactions will build complex molecules. Catabolic reactions will break down molecules into smaller ones. Animals store carbohydrates as glycogen, while plants store carbohydrates as starch. So choice B. So just remember, animals glycogen and plants starch. So glycerol is just a part of fatty acids. Cellulose is a component of plant cell wall. So those choices have nothing to do with this. Number 18, when different types of macromolecules are available for ATP synthesis, they will be used up in which order? So first we'll do carbohydrates, then fats, then proteins, because carbohydrates are most efficient and the easiest for cell to put through cellular respiration. Fats, we can do that too, but it's more complicated. And then proteins we need for other purposes. So we will only use proteins for ATP synthesis if we have absolutely no other carbohydrate or fat molecule that we can use for this purpose. True or false questions, 2 points each. Oxidation is a loss of electrons. Yes, that's true, right? The reduction is when we gain electrons. The way you can think about it is that when we gain electrons, we gain negative charge. So we become reduced, right? So oxidation is a loss of electrons. A reduction is when we gain electrons. Oxygen is not very electronegative. That's false. oxygen is very electronegative. That's what makes it a very good electron acceptor during cellular respiration. Most cells have emergency storage of ATP enough to last for 30 minutes. That's false because ATP is not very stable. It breaks down very quickly, so we can't really have any storage of ATP. What we can do, we can have mitochondria constantly working to generate ATP that will be used very fast. Glycolysis begins with energy investment phase and ends with energy payoff phase. That's true. At the beginning of glycolysis, we use up 2 molecules of ATP, and then during energy payoff phase, we generate 4 molecules of ATP. So we end up with net gain of 2 molecules of ATP. As a result of pyruvate processing, pyruvate is converted to coenzyme A. So it's not true coenzyme A is actually the enzyme that does the work of pyruvate processing. And the final product of this reaction is acetyl-CoA. So that's not true, that's false. Citric acid cycle can be turned off at multiple points depending on the abundance of ATP. That's true. There are multiple points where it can be inhibited if we have too much ATP, so that we don't do anything that's not necessary. Cells are very, very efficient. Electron carriers such as NAD plus and FAD carry protons on top of electrons. That's true, right? Because if you think about it, NAD plus that's loaded with electrons becomes NADH. FAD that's loaded with electrons becomes FADH2. So that's because they carry protons or H plus hydrogen ions in addition to electrons. Citric acid cycle occurs in the intermembrane space in the mitochondria. That's false because citric acid cycle will take place in the matrix of mitochondria. Electron transport chain takes place in the inner membrane and cristae of mitochondria. That's true. And the intermembrane space of mitochondria has a lot of positive charge during cellular respiration. That's true because that's where we will pump all the hydrogen ions. And that's how we build that electrochemical gradient. And that's why, because they're positively charged, we will have a lot of positive charge there. Okay, matching 2 points each. Don't forget to record right answers, score your points as we go along. So number 1, feedback inhibition is either we have a definition here or maybe we have an example of it. So that's GATP binds to a regulatory site of phosphofructokinase, then the reaction slows dramatically. That's an example of feedback inhibition that happens during cellular respiration. Feedback inhibition is when, let's say, we have a reaction that happens inside cells and it produces some product. So sometimes we can have too much of that product and we don't need that reaction to happen anymore, or we need to slow it down, right? So either we need to slow it down or we need to stop it from happening at all. So the product will come and it will bind to the regulatory site of the enzyme that's carrying out this reaction, and the reaction will either stop or it will slow down. So that's a way for cells to be very efficient and only carry out reactions that are necessary. Fructose 6-phosphate. So fructose is a 6-carbon sugar, right? So let's look at fructose. I'll just do a quick Google search. So that's fructose. If you look at it, it's very similar to glucose. And because it's very similar to glucose, it must be some molecule that is part of initial processing of glucose, right? So it's part of glycolysis. Let's see, and it fits choice D, right? An intermediate molecule of glycolysis. Number 3, cristae, that's those folds that are found inside the mitochondria. So sac-like structures inside the mitochondria. ADP is formed when an outermost phosphate group breaks off from the precursor molecule. So the precursor molecule is ATP. So the way we form ADP is ATP has 3 phosphate groups. We will break off the outermost phosphate group, and this is how we will form ADP from ATP. So that's choice A. NADH is an electron carrier, so that's choice J. NADH or FADH2. Those are electron carriers. Oxaloacetate, that's an intermediate molecule in citric acid cycle. So why am I doing this, like fructose 6-phosphate, oxaloacetate? The reason I put those choices there is because your teacher won't expect you to know all the intermediate molecules because there are a lot of them and they're not so important. But you need to be familiar with the process. And if you're familiar with the process, you will be able to recognize those intermediates, right? So you won't be able to list all those intermediates in the proper order, but at least you should be able to recognize them. And then phosphorylation is when molecules changes shape after addition of a phosphate group, right? So phosphorylation comes from the word phosphate. So we add phosphate group, molecule will change shape and will do some work in the cell, participate in some chemical reaction. Chemiosmosis is when ATP is produced via a proton gradient. That's E. And ubiquinone is a component of electron transport chain. That's F. So that's one of those molecules that are part of electron transport chain. It's a very mobile molecule. So other molecules that are part of electron transport chain, they're just embedded in the membrane. Ubiquinone can move. So that's why it's very important. It accepts electrons, but it also transports them. And then 10, we're left with choice C, potential electron donors for some prokaryotes instead of glucose. So some prokaryotes won't have access to glucose necessarily or won't be able to metabolize it. So they will use H2, CH4, or H2S for that purpose. So number 5, short answer question, that's 6 points. Describe how electron transport chain works. Use words such as inner membrane, intermembrane space, matrix, electrochemical gradient, ATP synthase, and protons. I wrote down the answer here because I didn't want to take the time to type it now. So citric acid cycle happens in the mitochondrial matrix, while the electron transport chain is located in the inner membrane. So if we look at mitochondria has 2 membranes, right? So this is our mitochondria. So this would be the outer membrane. This would be our inner membrane that has a lot of those folds, right? Sacs, cristae. And then we have the intermembrane space. I will color it green. So all the green stuff is the intermembrane space. And it's really, really, really important for cellular respiration. So what happens? So the citric acid cycle will happen here in the matrix, right, inside, while the electron transport chain will be located in the inner membrane, which is here, right? This is the inner membrane. That's where electron transport chain will be embedded. As electrons go down the electron transport chain, they lose energy, right? It's like a staircase going down. Every time they go down, they lose energy. And that energy is used to transport protons across the inner membrane into the intermembrane space. So now we have a lot of protons build up here in the intermembrane space. So protons are positively charged, and accumulation of them will create an electrochemical gradient in the intermembrane space, right? We have a lot of positive charge in one place, and we have a lot of the same kind of chemical in the same place, right? So that creates an electrochemical gradient because those protons, they are repulsed by each other. They have the same charge, so they experience repulsion. They also want to go down their chemical concentration from the intermembrane space out. They want to diffuse, so they will flow back into the matrix. So there are those enzymes called ATP synthase, and they are embedded in the inner membrane. So those protons, they will go down ATP synthase, and the energy that is released from that process will be used to make ATP. So this is how it works. If you got part of it correct, you can give yourself partial credit, or if you describe this process correctly, of course, give yourself full credit. So now there's bonus question. Contrast substrate level phosphorylation and oxidative phosphorylation. When does each happen during cellular respiration? So this is just from my experience teaching. I discovered that this is two terms that give students a lot of trouble. They do sound kind of complicated, right? Substrate level phosphorylation, oxidative phosphorylation. But what does it really mean? So phosphorylation, we just add a phosphate to ADP, we make ATP. So there are two ways it can happen. It can happen through enzyme, right? So enzymes take some molecule, and they work with it. They speed up chemical reactions. So the molecule that enzymes work with is called substrate. So substrate level phosphorylation with enzymes will happen during glycolysis and citric acid cycle, when enzymes phosphorylate add phosphate group to ADP. Sorry, it should say ADP, my mistake there. Oxidative phosphorylation happens during electron transport chain and chemiosmosis, when phosphorylation of ATP is coupled with oxidation of electron carriers. NADH and FADH2 will come in, and they will unload their electrons, and they will go down electron transport chain, and that's what will allow us to have this phosphorylation, right, to produce ATP during electron transport chain. So when we unload electrons, electron carriers lose those electrons. So that's why we call it oxidative, right? Because oxidation is a loss of electrons. Electron carriers lose electrons, they become oxidized. So that's why production of ATP during electron transport chain and chemiosmosis is called oxidative phosphorylation. So that's it. Please score your points. If you got a good grade, congratulations. If you didn't do so well, good job for trying, and now you learned something. So now you know this material better, so if you have to do tests on cellular respiration again, you'll do much better, right? So it's all learning experience. So you checked your knowledge and you learned something new, you improved your knowledge of this material. So that was the whole point. If you have any questions, please mention them in the comments below. If you enjoyed watching this video, please give me a like and subscribe so that you don't miss future content, because I'll be coming out with more practice tests like this. I post practice tests on different topics in biology and chemistry. So my chemistry practice exams will have blue thumbnails, and my biology practice exams will have green thumbnails. Just to make it easier to find them in my channel. So hopefully see you next time. Bye bye.